Welcome back, friends. Welcome to another part of the digestive system lecture. That's the third part of the digestive system. And here we are talking about the different types of digestive system secretions. There are different organs present in the digestive system. Those are made with different tissues and cells. They are destined to produce a specific type of chemical factors that helps in the process of digestion. Digestion deals with both secretions as well as absorptions. First, they need secretions to break down the food into smaller fragments and produce nutrients in the unit form. Then they will take those nutrients and absorb that. So digestion is destined and designed in such a way. Now let's look at the process of the secretion. And the digestive secretions involve the production and release of juices and hormones by the gastrointestinal tract and its accessory glands like liver, pancreas, gallbladder and stuff. The goal for our learning is to understand and list the secretion of the digestive tract, to describe their function and to describe the control of the secretion with the help of both nervous system as well as hormone mediated system. The first slide that I want to talk about is more of a fact slide where we want to say the typical daily consumption of food is approximately 800 gram for an adult and fluid is approximately 2 liter. Now among that, about 1.5 liter of the saliva is secreted into the mouth per day. So just imagine those things, 1.5 liter of saliva, approximately 2 liter of gastric juices secreted every single day, and pancreas delivers more 1.5 liter of the pancreatic juice, while liver or gallbladder start producing the bile of 0.5 liter, that's a half liter, and small intestine produces approximately 1.5 liter of the fluid as well. So you think that this whole secretions, if we join them together, it approximately gives us around 9 liter. So it's a huge deal, right? So the gastrointestinal tract contains about 9 liter of the fluid every single day, and it's about only 0.15 liter is eliminated with the feces. So most of them are required for the conversion of the food that we take and then we ultimately absorb in our body, and which is very remarkable, and that help us to understand about how remarkable our digestive system really is. Let's move into the first kind of secretion, that is salivary gland secretions, sorry, it should be gland or saliva secretion. The extrinsic salivary glands include 3 different types. One is a parotid gland, this is a pair of parotid gland actually, submandibular gland and sublingual gland. These are the 3 major types of glands that we have as a salivary gland. While parotid glands produce serous fluid, and that fluid contains enzymes, electrolytes and different kind of mucin, a little amount of mucin which helps in to produce mucus a little bit. That's why we have a kind of mucus around in our mouth. Submandibular gland and sublingual gland produces more of a viscous fluid than parotid gland, okay, and all of them ultimately releases whatever things we call them saliva that carries salivary amylase enzyme that has the capability to break down starch and polysaccharide. So that's why if you eat any carbohydrate food, there is a first kind of chemical reaction will take place in the mouth due to the release of all the saliva coming in from the salivary glands. So saliva provides plenty of functions. One of the major function is the protection of our mouth against different bacteria and other foreign pathogens, because they have an antibacterial molecule secreted known as lysozyme. It's present in our saliva. It also carries immunoglobulin A, that is an antibody targeted, and this immunoglobulin A is known as a secretory antibody because it's secreted with saliva, tears and stuff. So that helps in protecting our body. Saliva also helps in the process of taste because it will dissolve the food chemicals. Then it will also help in the lubrication, right, because it will also produce a mucus a little amount. And fourthly is a digestion that we talked about, where it can break down starch with the help of amyls. Now if you look at the regulation of the saliva secretion, it is regulated both with the help of mostly it's with the help of the nervous system. The whole process is regulated there. So there are two scenarios you can see here. One is the satiety and then the stress. So normally when there is a process of involving with eating a food, or you're seeing a lot of foods and stuff here around you, and you have the access to all of them, so the parasympathetic nervous system of your body start producing acetylcholine. Acetylcholine helps involved with some chemical signaling inside the cell that ultimately produces inositol triphosphate and diacylglycerol. Both of them are kind of, diacylglycerol is a part membrane bound structure that will ultimately allows the further downstream signaling, which helps in release of and fusion of the saliva containing contents and vesicles inside the cell, that allows the saliva to come out in your mouth. While if you are going through any kind of stress, then sympathetic nervous system will start acting and norepinephrine will be made, that will produce a lot of cyclic AMP, and once they produce cyclic AMP, that will prevent the release of saliva. So mostly the process of salivation is controlled by autonomic nervous system, both the parasympathetic and sympathetic, as we saw just now. And normally they involve with the nerve, facial nerve, that's nerve number 7, hope I am correct, and another one is the glossopharyngeal nerve, that is the nerve number 9 here, glossopharyngeal. Both of these nerves carry this whole process. This parasympathetic stimulation causes watery and enzyme rich secretion from the salivary glands. Normally the touch, sight and the smell of the food stimulates these salivary glands, and you can just take different experiments to prove that. We have plenty of experiments to prove that thing. Like if you are entering into a good restaurant with a very appetizing smell, you see the appetizing smell, that means that smell stimulates our sympathetic and parasympathetic, mostly parasympathetic nervous system influence to produce saliva. And acidic substances and pressures of chewing also cause the increase of the release of saliva, because if you put something in your mouth and start chewing it with your teeth, that will also allow to release more of a saliva from the salivary glands. On the other hand, if you have a stress, kind of fear or fatigue, or if you're sleeping in these conditions, that inhibit the process of salivation. Otherwise, just imagine the amount of saliva we produce while we are staying awake is generally not produced during sleep, but sometimes while you are looking through a dream or something like that, saliva sometimes come out during the sleep as well. And second thing, that is another very important kind of secretion that goes on mostly in esophagus, is known as secretion of mucus, and that's also we can call it as esophageal secretions. And this is the only secretion that is made by the esophagus, okay, because this mucus is very important component, and they actually produce component known as mucin, which is converted into mucus. Mucus is a very viscous and mobile thing that will actually make this area of esophagus slippery, and it also coats the whole esophageal cells so that the food can easily pass through the esophagus. This is one thing. Second thing is that normally esophagus is connected with the stomach, so if there is, stomach starts secreting acids in different contents, and there is a chance if stomach start to squeeze back and the lower esophageal sphincter is somehow releases, these acids can backflow into the esophagus. This phenomena is known as acid reflux, or it's also known as a disease called GERD, chronic esophageal reflux disease. That thing can cause the esophagus cells to kind of damaged by the activity of the acid, but the content of acidity is very high here in the stomach. So to prevent that, this esophagus produces those mucus that covers the whole esophageal wall and protect us against it. So this is how it look like. This is the intestinal fluid, these are the cells, and this will start producing bicarbonate to fight against that acid as well, because bicarbonate as well as they start to neutralize the environment of pH, because stomach lumen have a pH around 2, which is very acidic, like the hydrochloric acid and sulfuric acid at that range. Normally they produce hydrochloric acid, but that's very acidic, this pH 2. So once they produce, this mucosal cells arranged to have a based on pH 7, and they start secreting those things that to protect us against it. Now if you go to the third kind of secretion, it is a gastric secretion. This is one of the major secretions of our digestive system. Gastric secretion is like mostly it's called a gastric mucosa. Those are the cells that are present there. They start producing different chemicals with the help of exocrine and endocrine as well as paracrine secretions. The exocrine secretions collectively called the formation of gastric juice. Approximately 2 liters of this gastric juice is produced every single day, and it includes mucus. So generally gastric juice has several constituents. I say it has mucus, it has HCl, it has pepsinogen. Pepsinogen is a type of enzyme that can produce pepsin. Actually pepsinogen is a precursor of the enzyme pepsin, which is a protease enzyme, and they start to produce several type of intrinsic factors which has functions like mucus throughout the stomach is produced, and it's protecting the lining of the stomach cells. Pepsinogen is also secreted throughout the stomach. HCl is produced by fundus and body. These are the two parts that produces HCl through this place. Okay, and intrinsic factors are also produced by fundus and body. Okay, but rest of the parts produces rest of the body place. And this is signaling process known as enteroendocrine signaling, because if you look at the cells now, if you zoom into the structure, you'll see these are the zoom in structure. This is another same kind of structures we are observing here. Here you see different cells like G cells are present. These are the G cells. There are also chief cells, there are parietal cells, there are mucus cells. Different cells are present, and some of those cells known as enteroendocrine cells. Those enteroendocrine cells that are mainly present in the pylorus region, these are where the enteroendocrine cells are present. They start releasing gastrin. Gastrin is a hormone, and that's why we call these cells enteroendocrine hormones, enteroendocrine cells, because those are endocrinal secretions and secreted by the cells that are present in the enteric body. And here you see that all of them, right, chief cells, mucus cells, they start releasing components and factors inside by that fashion. The specialized cell produce each of these gastric secretions separately. For example, the stomach mucosa is invaginated, if you look, because you know this whole stomach body, this whole cells are invaginated and they form deep wells they are called as a gastric pit, and they are filled with gastric glands, as you see here. If you look at the fundus, this area, you'll see they start forming these invaginations, and one of such invagination is this one forming called gastric pit and gastric glands. Gastric glands produces mucus, because if you zoom into one of the gastric glands, it has mucose cells, they produce mucus. Gastric glands have like in the body as well as in fundus, they also produce HCl, and there are two types of mucus that are produced in the stomach. One is the thick alkaline mucus, another one is thin watery mucus. The parietal cells, if you look at here, these are the parietal cells, the specific design cells in this picture. These parietal cells, they start producing HCl and intrinsic factors, while mucus cells producing mucus. And there are other cells known as chief cells. These pink colored cells are the chief cells. These chief cells, They are also known as zymogenic cells, produce zymogen, which is an inactive proteolytic enzymes. Okay, and that will help to produce pepsinogen. And some cells in the gastric glands of the pylorus, if you look, they have a G cell. G cells mainly present in the pylorus, they are enteroendocrinal cells, they produce gastrin. Okay. Although all of this, most of the secretions are made from the stomach cells and the gastric patent gland that is formed inside the stomach, but we still keep another slide for explaining the stomach secretions and some examples of that as well. Normally, we know that stomach produces mucus, it produces HCl, intrinsic factors and stuff. The mucus that stomach produces, that protects the stomach from the self digestion, that's also known as auto digestion by all those neutralizing stomach acids that's present there. So here this mucus actually neutralizes those stomach acids, and also it inhibits the activity of the protease known as pepsin. While we have mucus and the tight junctions between the cells in the gastric mucosa, they are collectively known as the gastric mucosal barriers, because if you look at the cells that are present and making the stomach, it is not loosely placed like that. We have a tight junction formed in between the cells so that all those acid contents and all those components cannot take entry in between the intercellular junctions there. Okay. Only aspirin and alcohol are absorbed across the stomach's mucosal epithelium. Otherwise, rest of the things don't have any entry point there. When aspirin or alcohol passes through the stomach's mucosal barrier, they can destroy those cells. That's why ingesting a lot of alcohol or aspirin day by day can harm your stomach, in turn harm your digestive system. And the HCl that is secreted by the stomach, especially the parietal cells, that lowers the luminal pH between approximately 1.5 to 2, this pH content that is produced. Mostly it's produced by fundus and the body. You see the proton content is listed here, and in this case this change in pH, like on the scale that represents 10-fold change in pH in the stomach, for the example of 2 pH, that is compared to the mouth, which is 7. It's 2 pH compared to the mouth, that is 7. So imagine how much acidic the content will be here. And actually the release of HCl is not to digest a food, it's actually to make other enzymes activated and that high acidic content. For making all those proteolytic enzymes active, they need to release this acid. The highly acidic gastric environment is also very lethal for most of the bacteria and other microorganisms. So we won't find generally bacteria there in the stomach until and unless there is any compromisation in the lining production in the stomach. And if this compromisation is done due to the taking of aspirin or taking of a lot of alcohol day by day, or due to any kind of ulcer formed there, then bacteria can take place or take entry through that place. Or there are few bacteria like Campylobacter jejuni, they can actually present there in the stomach, and they can actually be there beating this acidic content over there. So that's one kind of bacteria that can colonize in stomach even though there is a high acid content, but most of the bacteria cannot. The HCl functions here to break down the plant cell walls, most of the connective tissues if we eat the plant product, because we used to eat a lot of plant products there. It denatures the protein a little bit, but mostly it converts that pepsinogen, which is the precursor molecule of pepsin, into the active pepsin. Once it produces active pepsin, that is the active enzymes to break down protein components, right? Now the parietal cells start secreting another factor known as IF, or intrinsic factor, which is also required by the intestinal cells to absorb vitamin B12. If we don't have intrinsic factor released properly in our stomach, vitamin B12 absorption will be halted, because if the vitamin B12 content is halted, if we can't absorb vitamin B12 in our body through stomach, that means our RBC production will be compromised there, because production and maturation of RBC required a lot of vitamin B12, because it's a cofactor. This acts as a cofactor in the process of the RBC maturation, and that can cause anemia known as pernicious anemia. The peptides in the stomach triggers the release of another hormone called gastrin, that is from this pyloric region, and these are the cells present here known as the G cells, through where all these molecules, all these hormones start to release. And they also, this gastrin stimulates the HCl secretion by the parietal cells, and also the histamine secretion by the paracrine cells that is present here. So paracrine cells also produce these histamines, and you know, histamine acts as a kind of a it synergistically act with gastrin to stimulate the release of HCl. So it's kind of a positive feedback loop going on there. Once it produces this gastrin from the G cell, it start producing more histamines from as well. So histamine and gastrin together help producing more and more HCl in the stomach to make the content more acidic and keep the content acidic over the time. That's the idea. Now if you go to the other type of secretion made by pancreas, it's known as the pancreatic secretions. Pancreatic secretions mean they produce a lot of juices and chemical factors together, known as pancreatic juice. Now most chemical that produced by the pancreatic secretions, they are required in the digestion process, and mostly they are proteolytic, and also a few parts they also produce lipolytic enzymes as well, but very less, mostly proteolytic. The secretions that initiate the process of chemical digestion in the intestine actually comes from the pancreatic juice. Okay, and in this case they produce some important facts. One of such important fact is bile, that produced by the liver mostly, but it is stored and released from the gallbladder, that emulsifies fat to increase their surface area, and subsequent chemical digestion will take place. While the exocrine pancreas produces two types of pancreatic juice: enzyme rich pancreatic juice, that is one thing, enzyme rich, and second one is bicarbonate rich. So one of such juice contains a lot of enzyme factors, one of such contains a lot of bicarbonate factors. Both of them are required in different purpose. The enzyme rich pancreatic juice is stimulated by the hormone cholecystokinin or CCK, while the bicarbonate rich pancreatic juice is stimulated by the secretion of secretin. Okay. Now the exocrine pancreas secretions are often delivered through the different sort of hepatopancreatic sphincter that is present there between the pancreas and the junction between the duodenum. So that sphincter regulates the release of those exocrine materials, and they follow with some of the enzymes. If you look at the enzyme rich secretion, they contain some of the enzymes. One of such is mostly proteases. Example of such protease is chymotrypsin, produced by the pancreas. Second type is amylase, that's known as a pancreatic amylase because it's secreted by pancreas. The first sort of amylase released by our salivary gland is known as salivary amylase, and it also produces lipase or lipase, whatever you say, the enzyme to break down lipid. Now the action of lipase will only be strong when there is a release of bile from the liver, otherwise lipase cannot act on large fragments of fat. Now pancreatic proteases includes chymotrypsin, trypsinogen, and procarboxypeptidase. All these things, chymotrypsinogen, trypsinogen, whenever we have a gen attached as a suffix there, that means that chemical or that enzyme is not properly active. These are the pre-molecules. We need to cut, and some sort of modifications are required to make them active. So let's say trypsinogen is converted into trypsin, chymotrypsinogen is converted into chymotrypsin, and procarboxypeptidase is converted into carboxypeptidase. And actually enterokinase is the enzyme that helps in the conversions of all those trypsinogen into trypsin, chymotrypsinogen into chymotrypsin and stuff. Once they produced, mostly like say the enterokinase enzyme that converts the trypsinogen into trypsin, enterokinase, that's the enzyme that converts chymotrypsinogen into trypsin. This trypsin activates chymotrypsinogen into chymotrypsin, and then trypsin activates procarboxypeptidase to produce carboxypeptidase. So these are chain events, but first started with enterokinase enzyme. Okay, and there are also a different set of cells known as duct cells that are also present inside this pancreas. Those duct cells start secreting bicarbonate. That's the second part of the secretion, bicarbonate rich secretion. This bicarbonate rich secretion from the duct cells to neutralize the acid from the stomach and to produce an optimal pH in the duodenum so that the rest of the process can work finely. Because stomach is designed in a way to support and live in the acidic environment, while the duodenum cannot receive that much of acid and live healthy. So duodenum needs to neutralize all those components, and that is done with the help of the secretion of bicarbonate there. Okay, and the endocrine pancreases, that's the process of endocrinal signaling. It also secretes some hormones because that's also famous for secreting hormones. Two such hormones secreted by pancreas, one is insulin, another one is glucagon, which are required for the metabolic pathways of after the digestion event, but not exactly in the digestion event. While insulin regulates the absorptive state and glucagon regulates the post-absorptive state of the process of the digestion, but mostly controlling the glucose concentration in the bloodstream, and that's an important role to play. The regulation of pancreatic secretions are made up with different hormones like secretin, GIP and CCK. Secretin says like acidity in the intestine causes increased sodium bicarbonate release to neutralize the environment. Fatty acids and sugar causes the increased insulin release, and CCK, fats and proteins, cause the release of this CCK. So all these things, depending upon what kind of content that we have and that we are digesting, stimulates the release of all those different hormones. And here is a feedback loops presented, a positive loop and a negative loop, where it's controlled by hormones as well as parasympathetic nervous system. Now it's time to look for the secretion comes from liver. And when you talk about liver, I always get fascinated about the function of liver, because this is one of the most important organ of our body, because it carries a lot of functions for metabolism as well as in digestion in so many different directions of our body, to release all the toxic materials from our body and everything. So this is kind of a hero organ of our body. The main digestive function the liver plays is to produce what is called as bile, and bile is associated in the digestion of fat. If they don't secrete bile, the fat digestion will be kind of impossible. The bile backs up into the gallbladder, so it's secreted by liver, and through this duct it will go to the gallbladder through this systolic duct, and finally those all ducts, common hepatic duct and systolic duct, they all tie together to finally form what's called a common bile duct that will ultimately release all those components into the duodenum. And they have here what is called hepatopancreatic sphincter just between this duct end, where we have the connection between the bile storage site, that is gallbladder, pancreas and the liver. So three different ways connected to one single common duct. There is a sphincter to regulate this whole process, that is hepatopancreatic sphincter. Now the two major components of bile is organic compounds and bicarbonate solutions. The organic compounds that we have mostly are bile salts that helps to emulsify the fat. So if you have a large fat component, it will break those components down into smaller chunks and fragments, so that all those lipolytic enzymes have much more surface area to work and break down those fat components into further smaller fragments, right? And bile work associated with the pancreatic lipase, which comes through the pancreas. So once bile fragmentizes and emulsifies those fat, then lipase will act and break the fat down into fatty acids and glycerol. That's how the digestion takes place there. Okay, and it also contains lecithin, it contains cholesterol, and it contains bilirubin. So bilirubin, lecithin, cholesterol, and all the rest of bile salts are secreted there. While cholesterol and bilirubin are eliminated through the feces, but bile salts are recycled throughout the time because they are required all the other times in different process. They are recyclable, and they stimulate the secretion of bile from the liver, and then those bile can store into the gallbladder. So that's why we have a storage site, because liver is the busy organ, it cannot produce those bile wherever it's required in their process. So whenever you digest food, some part of the bile which is stored in the gallbladder, that will come to help in the process of digestion. So when we stimulate this whole process, like when you stimulate the release of these chemicals by the enzyme, by the hormone actually, secretin, that's called as nature's antacid. Secretin is secreted. Bile duct cells secrete bicarbonate solution that is identical to the pancreatic bicarbonate solution as well, and that function as a liver neutralizing agents. So here liver is not only allowing the fat digestion, but also it starts secreting all those enzymes. It starts release that secretin, and secretin, remember this hormone known as a nature's antacid, because it's naturally protecting us against acidity, because it's a bicarbonate solution that's produced. Now it's time to look at the secretions made in the small intestine. Small intestine have the submucosal glands, it has the Paneth cells, it has the goblet cells. All those different types of cells and tissue have different secretions. While the goblet cells release mucus, Paneth cells produce enterokinase, which is an enteric kinase enzyme that activates all those pancreatic zymogens, and submucosal glands produce viscous mucus to prevent the damage of the mucosa. Okay, if we look into little deep, we have those intestinal glands, or also known as crypts, intestinal crypts. Here you can see the detail of those such of a intestinal crypt, where you have a brush border actually made up with the villi kind of cytosolic extensions of those cells in the intestinal epithelia. It's like a brush border, because those are the microvilli of the cells that we can see. And the concept of villi and microvilli is that normally the cell lining should be like this. Once they form villi, it start to form like this. Once it form microvilli, it actually start to form things like this. So start to think of why we need to do these things. This is without any villi, this is with villi, without microvilli, this is with microvilli and villi. So the more we are dividing, that means the more surface area is increasing, and the job of the small intestine is the digestion as well as absorption of the food. That should be placed properly here. So that's why we have a lot of microvilli contents produced in there. Okay, and those cells, those epithelial cells, they produce certain enzymes. Obviously that's one part of the job, but they have another job of absorption. So that's why they need to keep in mind to modify all their components so that they can interact and involve with this whole process. Lastly, we have a large intestinal secretions. Large intestinal secretion, they have alkaline mucus in the large intestine containing bicarbonate. The mucus that we have in the large intestine, we have a bicarbonate, we have potassium ion that's secreted by the intestinal cells. because both these potassium ions that is secreted as well as this bicarbonate helps in the neutralization from the stomach acids and chyme. Potassium also helps in the process of preventing any kind of bacterial acids and damage. The role of mucus in the large intestine is much bigger compared to the any part that mucus is present, because the mucus in the large intestine coats all the mucosal cells, all the epithelial cells, that the first hand in the first order is coating all the cells, so it will prevent any sort of acidic damage. It will also prevent any sort of pathogen to enter, because this is large intestine and this is filled with E. coli. It's a healthy and friendly bacteria of human body that resides there. Not only E. coli but multiple bacteria resides there in the intestine, and the role of them is to produce vitamin K, and they also fight against some other invaders or foreign pathogens, right? That's why we need to take a like close look of gaining a good healthy gut microflora is a better way of living healthy life. That's why we involved with the idea of probiotics, where we take all those healthy bacteria from outside with the help of like yogurt and other stuff, so that it can keep us healthy. Now the mucus that we see here, it lubricates the feces and protects the large intestinal wall from any sort of mechanical damage and friction that is generated during the release of the feces. Let's say a person is not drinking adequate water. In that case, the feces becomes hard and very hard and strong. That can lead to the damage in the intestinal wall, that can ultimately lead to the release of blood and stuff thereover. That can be prevented. And both the reflex that is required here, long reflex as well as short reflexes that are triggered by the mechanical stimulation of the acid and the increased secretion of alkaline mucus from the large intestine start to release. So the more friction it will be generated, the more feces are going to involve with the interaction with the wall, the more they want to create mucus, so that those feces will remain soft and it can release without harming any of the cells and intestinal lining of our body. That in a sense are different sort of secretions that are required in the process of the digestion. So I hope this video helped you to understand about the digestive secretions. If you like this video, please hit the like button, share this video with your friends, and definitely subscribe to my channel to get more and more physiology videos like that. Thank you.